What if the story of a people was written not in books, but in their blood? The Ganges Delta holds such a tale, a silent history embedded in the DNA of the Bengali people. Have you ever wondered why their genetic map tilts towards the east, hinting at a forgotten migration? Journey with us as we unravel a story of conquerors, ancient diseases, and the surprising genetic unity that defines one of the world's most populous cultures. The Ganges Delta, a sprawling, fertile landscape shaped by the silt of two mighty rivers, has for millennia been a cradle of civilization. Its history written not only in the ruins of ancient cities and the verses of its poets, but also deep within the very cells of its people. This vast alluvial plain has silently preserved the genetic memory of the Bengali people, a complex tapestry woven from the threads of countless generations. It is a memory that extends beyond the reach of recorded history, a biological archive embedded in their DNA. This genetic legacy is not merely a product of inherited culture or shared language, but a direct consequence of the region's unique geography, which has made it both a sanctuary and a crossroads. For centuries, the Delta's rich agricultural lands and strategic waterways have attracted a diverse array of newcomers. Traders, seafarers, and adventurers from distant lands arrived, their ships laden with goods and their minds filled with ambition. They came from the maritime networks of Southeast Asia, from the overland routes connected to the heart of the Indian subcontinent, and from even further afield, each group leaving an indelible, though often subtle, mark on the local population. Conquerors, too, have swept through this land, their armies marching across the plains and their dynasties rising and falling like the tides. From the Mauryans and Guptas to the Turkic sultans and Mughal emperors, each wave of conquest brought new peoples and new genes into the deltaic melting pot. Yet, the story encoded in the Bengali genome is not simply one of replacement or domination. Instead, it speaks of a remarkable capacity for assimilation and synthesis. The newcomers did not erase what came before. Rather, they became part of a continuously evolving human landscape. Their genetic contributions were absorbed into the existing population, creating a unique blend that distinguishes Bengalis from their neighbors. This process of admixture was not a single event but a slow, continuous process, a quiet intermingling that occurred over hundreds, even thousands, of years. The Delta's environment itself played a crucial role in shaping this genetic narrative. The constant negotiation with the rivers, the floods that brought both devastation and renewal, the shifting channels that created new lands and submerged old ones, fostered a sense of resilience and adaptability. It was a landscape that demanded cooperation and interdependence, encouraging the breakdown of rigid social barriers and facilitating the mixing of different communities. The genetic memory preserved in the Ganges Delta is therefore a testament to this long and complex history of movement, interaction, and adaptation. It tells a story of a people who have been shaped by centuries of traders and conquerors, yet have forged a distinct identity that is all their own. It is a story that is not just about origins, but about the enduring power of a place to shape the people who call it home. The DNA of the Bengali people is a living chronicle of this past, a silent witness to the ebb and flow of empires, the voyages of ancient mariners, and the quiet persistence of life in one of the world's most dynamic landscapes. When viewed through the lens of population genetics, the people of the Indian subcontinent generally form a broad, interconnected cluster, a testament to shared ancient ancestries and millennia of interaction. Yet, within this vast mosaic, the Bengali people occupy a unique and intriguing position. On genetic ancestry maps, which chart the relationships between different populations based on shared DNA markers, Bengalis do not cluster neatly and exclusively with other South Asian groups. Instead, their genetic profile displays a subtle but consistent and unmistakable tilt toward the east, a clear indication of deep and ancient interconnections with the peoples of Southeast and East Asia. This eastward shift is not a mere statistical anomaly. It is a fundamental characteristic of the Bengali genome, a signature that sets them apart and speaks to a distinct historical trajectory. While the majority of their ancestry is undeniably South Asian in origin, this additional component provides a crucial piece of the puzzle in understanding who the Bengali people are and where they came from. The presence of this Eastern genetic influence challenges simplistic narratives of population history in the region, which have often focused exclusively on migrations from the northwest. It suggests that the eastern frontiers of the subcontinent were not a periphery but a vibrant zone of interaction, a porous boundary across which people, ideas, and genes flowed freely. This genetic tilt reflects ancient migratory streams and trade networks that are often overlooked in mainstream historical accounts. The Bay of Bengal was not a barrier but a bridge, connecting the Ganges Delta to the coastal civilizations of Myanmar, Thailand, and beyond. Similarly, the river valleys and mountain passes of the northeast served as conduits for the movement of people from what is now southern China and Tibet. These connections were not fleeting or ephemeral. They were sustained over long periods, allowing for a significant degree of intermingling and genetic exchange. The result is a population that is geographically located in South Asia but genetically and culturally oriented, in part, toward the east. This dual identity is a core element of the Bengali heritage. The subtle eastward tilt on genetic maps is the quantifiable, biological evidence of this long-standing relationship. It is a reflection of a history in which the delta was not an endpoint but a nexus, a place where different worlds met and merged. This unique genetic positioning underscores the complexity of identity in South Asia and highlights the importance of looking beyond traditional geographical and cultural boundaries to understand the true story of its peoples. For Bengalis, this genetic signature is a reminder that their roots run deep, not only into the soil of the Indian subcontinent but also eastward, across the hills and seas, connecting them to a wider Asian world. Genetic analysis allows scientists not only to identify the different ancestral components that make up a population, but also to estimate when these different groups came together. For the Bengali people, this molecular clock has pointed to a particularly significant period of admixture, a time when a substantial new genetic element was introduced into the population. Sophisticated statistical models, which analyze the length of shared DNA segments from different ancestral populations, have revealed that around 10% of the typical Bengali person's ancestry originates from East or Southeast Asia. This is not an insignificant trace amount, but a statistically significant contribution that has had a lasting impact on the region's genetic landscape. Crucially, the data suggests that this major infusion of new genes was not a gradual trickle over many centuries, but likely occurred around a specific point in history, approximately the 6th century CE. This period corresponds with a time of significant political upheaval and transition in the Indian subcontinent. The once mighty Gupta Empire, which had dominated northern India for centuries, was in a state of terminal decline. Its centralized authority had crumbled, leading to a power vacuum and the fragmentation of its vast territories into smaller, regional kingdoms. This era of instability and realignment created new opportunities for migration and settlement. As the old imperial structures weakened, populations on the peripheries of the empire began to move, seeking new lands and new opportunities. It is within this context of imperial collapse and demographic flux that the significant East and Southeast Asian admixture into the Bengali population likely took place. The timing, around the 6th century CE, provides a historical anchor for the genetic data, allowing us to connect the abstract world of DNA with the tangible events of the past. The decline of the Gupta Empire would have loosened control over the eastern frontiers, making it easier for groups from the northeast to migrate southward and westward into the fertile plains of the Ganges Delta. These were not necessarily large-scale, organized invasions, but more likely a series of smaller, uncoordinated movements of people, families, and clans. Over time, These migrations were substantial enough to introduce a new ancestral component that accounts for a remarkable 1/10 of the modern Bengali genome. This 6th century event was a pivotal moment in the formation of the Bengali people, a demographic shift that reshaped their genetic identity. It demonstrates that the story of Bengal is not one of isolation, but of dynamic interaction with its neighbors, particularly those to the east. The legacy of this period is written in the DNA of every Bengali person today, a permanent echo of a time of imperial decline and population movement that fundamentally altered the human geography of the region. The introduction of a significant East and Southeast Asian genetic component into the Bengali population around the 6th century CE raises a crucial question. How did this admixture occur? The historical and archaeological records of this period in Bengal are sparse, and they do not speak of large-scale invasions or military conquests from the east that would have forcibly replaced the existing population. The genetic evidence itself supports a different narrative. The pattern of admixture is not consistent with a scenario where a small, conquering elite imposed its genes on a much larger subjugated population. Instead, the data points toward a more peaceful and gradual process of integration, a slow blending of peoples over time. The most likely source of this genetic infusion was the various Austroasiatic and Tibeto-Burman speaking groups who have long inhabited the hills and river valleys to the northeast of the delta, in regions corresponding to modern-day northeast India, Bangladesh's Chittagong Hill Tracts, and parts of Myanmar. These communities were not part of a conquering army but were likely driven by a combination of factors, including environmental pressures, population growth, and the political vacuum left by the decline of the Gupta Empire. They did not arrive as a unified force but as a steady stream of migrants, moving into the delta and settling alongside the existing inhabitants. The process was one of cultural and social osmosis rather than violent subjugation. These newcomers brought with them their own languages, customs, and agricultural practices, which gradually blended with those of the local Indo-Aryan speaking population. Over generations, this cohabitation led to extensive intermarriage, the primary mechanism for genetic admixture. The boundaries between the indigenous delta dwellers and the northeastern migrants slowly blurred as families merged and communities became intertwined. This model of gradual integration is supported by the relatively even distribution of the East/Southeast Asian ancestry across the Bengali population today. It is not concentrated in a specific social class or geographical area, as might be expected in the aftermath of a conquest, but is instead woven throughout the social fabric. This suggests a widespread and democratic process of mixing, where the newcomers were not treated as a separate, subordinate group, but were incorporated into the evolving society of early medieval Bengal. This narrative of integration over conquest paints a picture of a dynamic and relatively open society, one capable of absorbing and assimilating new populations without resorting to systemic violence. It highlights the importance of the northeastern frontier as a crucial source of demographic and cultural innovation in Bengal's history, a corridor through which new peoples and new genes flowed, not through the force of arms, but through the more subtle and enduring processes of migration, settlement, and coexistence. The genetic makeup of the modern Bengali people is a complex palimpsest, built up over thousands of years through the layering of different ancestral populations. To understand this intricate composition, it is necessary to peel back these layers and examine the foundational elements that have contributed to the Bengali genome. At the very base is the oldest discernible genetic stratum, belonging to the indigenous hunter-gatherers of the subcontinent, often referred to by geneticists as ancient ancestral South Indians, ASI. These were the first modern humans to populate the region, arriving tens of thousands of years ago. Their genetic legacy forms the bedrock of nearly all South Asian populations, and in Bengalis, it represents a substantial and fundamental component of their ancestry. They were the original inhabitants of the delta, adapted to its unique ecological niches long before the arrival of agriculture or settled civilization. The next major layer was added with the arrival of pastoralists from the northwest, speakers of Indo-Aryan languages, whose ancestry is often termed Ancestral North Indian, ANI. This group, with genetic ties to the peoples of the Eurasian steppe and the ancient Near East, began migrating into the subcontinent several thousand years ago. Their arrival heralded a period of profound cultural and linguistic transformation, and their genes mixed with the indigenous ASI population, creating the foundational gradient of ancestry that characterizes much of South Asia today. In Bengal, this ANI component is a significant part of the genetic equation, linking the region to the broader historical and demographic processes that shaped the northern half of the subcontinent. However, the genetic story of Bengal does not end with this simple two-way mixture. The region's unique geographical position at the eastern edge of the subcontinent exposed it to different migratory streams, adding further layers of complexity. From the northeast came various Austroasiatic tribes, whose linguistic relatives are found today scattered across Southeast Asia. These groups were early agriculturalists who likely moved into the fertile delta plains, bringing new farming techniques and their distinct genetic markers. Their contribution is a key element that distinguishes the eastern parts of the subcontinent from the west, and their deep integration is confirmed by specific maternal lineages found in the Bengali population. Following them, or perhaps concurrently, were various Tibeto-Burman peoples, migrating south from the Himalayan plateau and the regions of present-day southern China. These groups brought another distinct ancestral signature, further enriching the genetic diversity of the delta. Each of these four foundational groups, the indigenous hunter-gatherers, ASI, the Indo-Aryan migrants, ANI, the Austroasiatic tribes, and the Tibeto-Burman peoples, contributed a unique set of genetic markers to the evolving Bengali genome. None of these groups exists in a pure form today. Instead, their legacies are interwoven, creating a composite identity. The Bengali people are the direct descendants of this multi-layered history, a living testament to the successive waves of migration and admixture that have defined the human story of the Ganges Delta for millennia. Decades of genetic research have allowed scientists to move beyond broad historical narratives and quantify the specific ancestral components that make up the typical Bengali genome. While individual variation exists, a consistent and characteristic pattern has emerged from numerous studies, painting a detailed portrait of the quintessential Bengali genetic composition. The dominant element, unsurprisingly, is of South Asian origin, a composite of the ancient indigenous, ASI, and later Indo-Aryan, ANI, ancestries. This component typically accounts for around 50% to 60% of the total genetic makeup. This places Bengalis firmly within the broader genetic landscape of the Indian subcontinent, sharing a deep and ancient heritage with their neighbors to the west and north. This South Asian foundation is the primary canvas upon which the unique features of the Bengali genome have been painted. What truly distinguishes the Bengali population, however, is the exceptionally high proportion of East and Southeast Asian admixture. This component, derived from the assimilation of Austroasiatic and Tibeto-Burman speaking peoples, is a defining feature of their genetic identity. On average, this Eastern ancestry constitutes a remarkable 20% to 30% of the Bengali genome. This is not a trivial amount. It is a substantial contribution that significantly alters their genetic profile compared to most other large South Asian populations. This high percentage explains the eastward tilt observed on genetic maps and is the direct result of the historical processes of migration and integration from the northeast, particularly around the 6th century CE. Furthermore, this proportion is not static across the entire Bengali-speaking region. There is a discernible geographical gradient. In communities located closer to the northeastern frontiers, in areas bordering northeast India or Myanmar, The percentage of East/Southeast Asian admixture can be even higher, sometimes exceeding the 30% mark. Conversely, in Western Bengali populations, further away from these historical migration routes, the proportion tends to be closer to the 20% end of the spectrum. This geographic variation provides a living map of historical migration, with the genetic signal being strongest near the points of entry. This typical composition, roughly 2/3 South Asian and 1/3 East/Southeast Asian, is the standard genetic signature of the Bengali people. It is a formula that encapsulates their unique history, standing at the crossroads of two of the world's major population zones. This blend is what makes the Bengali genome so fascinating to geneticists and historians alike, as it provides a clear biological record of the convergence of peoples and cultures in the dynamic environment of the Ganges Delta. The specific percentages may vary slightly from person to person, but the overall pattern of a dominant South Asian base with a powerful and significant Eastern infusion remains the quintessential and defining feature of the Bengali people. Given the complex history of migration and admixture that has shaped the Ganges Delta, one might expect the Bengali population to be highly genetically stratified and fragmented. The ingredients are certainly there: a foundational layer of indigenous hunter-gatherers, followed by Indo-Aryan migrants from the northwest, and significant contributions from Austroasiatic and Tibeto-Burman groups from the northeast. In many other parts of the world, such a history would lead to a population composed of distinct genetic subclusters, with clear genetic lines drawn between different social, religious, or geographical groups. Yet, when geneticists analyze the DNA of the Bengali people, they find precisely the opposite. Despite this incredible diversity of ancestral sources, Bengalis as a group show an unusually tight genetic clustering. This means that, genetically speaking, a Bengali Muslim from a rural village is remarkably similar to a Bengali Brahmin from a major city, and both are closely related to other Bengali communities, regardless of caste or creed. This genetic cohesion is one of the most striking features of the Bengali population and points to a unique social history. The primary explanation for this phenomenon lies in the historical social structures of the region. While the caste system, a rigid social hierarchy, has been a defining feature of many other parts of the Indian subcontinent, its influence in Bengal appears to have been less stringent, at least in a biological sense. The traditional rules of endogamy, the practice of marrying only within one's own caste or social group, seem to have been more porous and less strictly enforced in the Delta than elsewhere. This historical openness to intermarriage between different social strata allowed for a more thorough mixing of genes throughout the entire population. Instead of remaining isolated in separate genetic pools, the various ancestral components, ASI, ANI, Austroasiatic, and Tibeto-Burman, were able to flow more freely across social and religious lines, leading to a homogenization of the gene pool over time. The result is a population that is genetically much more uniform than its diverse origins would suggest. This lack of rigid long-term genetic separation is a powerful biological indicator of a social environment that, for long periods, facilitated a high degree of social mobility and integration. It suggests that while social distinctions certainly existed, they were not absolute barriers to human interaction and reproduction. This biological egalitarianism, as some have termed it, has played a crucial role in forging a strong, cohesive Bengali identity that often transcends other social divisions. The tight genetic clustering is the silent, cellular proof of this shared history of intermingling. It demonstrates that the story of Bengal is not one of permanent division and hierarchy, but rather one of continuous blending and synthesis, creating a people who are, despite their diverse roots, remarkably and unusually unified in their genetic heritage. The genetic makeup of a population is not shaped by migration and admixture alone. It is also profoundly influenced by the relentless pressure of the local environment through the process of natural selection. In the Ganges Delta, a region historically characterized by high population density and specific infectious diseases, these selective pressures have left a clear and fascinating mark on the Bengali genome, a story vividly told through the distribution of blood types. Globally, blood types O and A are the most common, while type B is relatively rarer. However, in the Indian subcontinent, and particularly in the Ganges Delta, the frequency of type B blood is significantly higher than the global average. This is not a random occurrence but strong evidence of natural selection at work. The prevailing scientific theory connects this high prevalence of type B blood to the historical presence of certain diseases, most notably cholera, which has been endemic to the Delta for centuries. The bacterium responsible for cholera, Vibrio cholerae, thrives in the warm, brackish waters of the Delta and has caused devastating epidemics throughout the region's history. Scientific studies have revealed a crucial link between blood type and susceptibility to this deadly disease. Individuals with type O blood, while having a robust immune response, are found to be significantly more susceptible to severe, life-threatening cholera infections. The cholera toxin appears to bind more effectively to the intestinal cells of type O individuals, leading to more extreme dehydration and a higher mortality rate. In contrast, individuals with type B blood seem to possess a degree of natural resistance. The specific antigens on their red blood cells may interfere with the bacterium's ability to cause severe disease, making infections less likely to be fatal. Over countless generations, in an environment where cholera was a constant threat, this simple biological fact would have had profound demographic consequences. In any given cholera outbreak, individuals with type O blood would have been more likely to perish before they had a chance to reproduce, while those with type B blood would have had a higher chance of surviving and passing on their genes to the next generation. This process, repeated over and over, would have gradually increased the frequency of the allele for type B blood in the population. The genetic landscape of Bengal was slowly but surely reshaped by the selective pressure exerted by a single microscopic organism. The high prevalence of type B blood among Bengalis today is therefore a living artifact of their ancestors' long and difficult struggle for survival in the unique environment of the Delta. It is a form of genetic adaptation, a biological memory of past epidemics, and a powerful example of how natural selection can mold the very essence of a people, right down to the antigens on their blood cells. The dense, waterlogged environment of the Ganges Delta, with its countless rivers, ponds, and rice paddies, has long been an ideal breeding ground for the Anopheles mosquito, the vector for one of humanity's most persistent and deadly foes, malaria. For millennia, the inhabitants of this region have been exposed to intense pressure from this parasitic disease, particularly its most lethal form, caused by Plasmodium falciparum. This constant, life-or-death struggle has driven one of the most well-documented examples of natural selection in the human genome, leaving a permanent mark on the DNA of the Bengali people in the form of specific hemoglobin mutations. Hemoglobin is the protein in red blood cells responsible for carrying oxygen throughout the body. Certain genetic mutations can alter the structure of this protein, and while these changes can sometimes be benign, they can also lead to inherited blood disorders. One of the most well-known of these is the mutation that causes sickle cell anemia. Another, highly prevalent in Bengal and Southeast Asia, leads to a condition called beta thalassemia. In a non-malarial environment, these mutations are purely detrimental, causing serious health problems for individuals who inherit 2 copies of the mutated gene. However, in a region where malaria is rampant, the story changes dramatically. Individuals who inherit just 1 copy of the mutated gene, making them carriers or heterozygous, gain a significant survival advantage. Their red blood cells, slightly altered by the single mutated gene, are less hospitable to the Plasmodium falciparum parasite. The parasite struggles to reproduce effectively within these cells, and the body is better able to clear the infection. As a result, Carriers of these hemoglobin mutations are much less likely to die from severe malaria. This protective effect created a powerful selective pressure. In every generation, individuals without the mutation were more likely to succumb to malaria, while those who carried a single copy of the gene were more likely to survive, reproduce, and pass that protective gene on. This heterozygote advantage led to a steady increase in the frequency of these mutations within the Bengali population, even though inheriting two copies resulted in a severe genetic disease. The prevalence of hemoglobinopathies like beta thalassemia in modern Bangladesh and West Bengal is the direct, albeit unfortunate, consequence of this evolutionary trade-off. It is a genetic adaptation that came at a cost. While these mutations offered crucial resistance against a deadly infectious disease in the past, they now contribute to a significant public health challenge in the form of inherited blood disorders. The presence of these genes in the Bengali population is a stark and powerful reminder of their ancestors' co-evolutionary battle with malaria, a biological scar left by the enduring mark of an ancient environmental pressure. While the analysis of the entire genome provides a broad overview of a population's ancestry, the study of specific genetic lineages can offer a more focused and detailed glimpse into the past. One of the most powerful tools for this is the analysis of mitochondrial DNA, mtDNA, which is passed down exclusively from mother to child. Because mtDNA does not recombine with paternal DNA, it preserves a clear and unbroken line of maternal descent, allowing geneticists to trace ancestry back through thousands of years. The specific variants of mtDNA, known as haplogroups, act as markers for ancient population groups and their migrations across the globe. When researchers examined the mitochondrial haplogroups present in the Bengali population, they found compelling evidence that corroborates the broader story of admixture told by the whole genome. The analysis confirmed the deep integration of Austroasiatic and Tibeto-Burman ancestries, particularly through the female line. Among the diverse array of mtDNA haplogroups found in Bengal, several stand out as being highly characteristic of East and Southeast Asian populations. For example, haplogroup M30 is found at its highest frequencies among Austroasiatic-speaking tribal groups in India, such as the Munda. Its significant presence among Bengalis is a clear genetic link to these ancient inhabitants of eastern India, who represent one of the key northeastern migratory streams into the delta. Similarly, haplogroup M4, while found across the subcontinent, has specific sublineages that are strongly associated with Tibeto-Burman speaking populations from the Himalayan region. The presence of these maternal lines within the Bengali gene pool points directly to the assimilation of women from these northern communities. These are not just abstract genetic markers. They represent the real-life stories of countless women who, centuries ago, migrated from the hills of the northeast into the plains of the delta, married into the local communities, and raised children, thereby weaving their maternal genetic legacy into the fabric of Bengali society. The persistence of these specific haplogroups, M30, M4, and others with similar origins, provides irrefutable, independent confirmation of the historical narrative. It shows that the admixture with northeastern groups was not a male-dominated process, as can sometimes occur in conquests, but one that involved the full integration of families, with women playing a central role. These maternal haplogroups are genetic echoes of the ancestors, faint but clear signals that have been passed down from mother to daughter for generations. They tell a story of migration, intermarriage, and assimilation, providing a powerful, female-centric perspective on the formation of the Bengali people and confirming the deep and ancient roots that connect the Delta to the diverse peoples of the Northeast. The story of a people's ancestry is recorded not only in their own DNA but also in the DNA of the trillions of microorganisms that live within them, particularly in the human gut. This complex community of bacteria, viruses, and fungi, known as the gut microbiome, is essential for digestion, immunity, and overall health. For most of an individual's life, their gut microbiome is relatively stable, and it is passed down, in part, from mother to child and shaped by close family contact and shared diet. Over generations, this results in distinct microbial signatures that are characteristic of different human populations. When scientists have studied the gut microbiome of the Bengali people, they have discovered another layer of evidence that reinforces the narrative of deep integration with populations from the northeast. The composition of the Bengali gut microbiome shows a fascinating blend of features. As expected, it shares many characteristics with the microbiomes of other South Asian populations, reflecting a shared dietary history centered on staples like rice and lentils, as well as exposure to a similar set of environmental microbes. However, just as with their human DNA, the Bengali microbiome exhibits a distinct shift toward patterns typically seen in East and Southeast Asian populations. This includes the presence and relative abundance of specific bacterial species that are more common in people from regions like China, Thailand, and Myanmar than they are in other parts of the Indian subcontinent. For example, certain species of Bifidobacterium and Prevotella, which are highly adapted to digesting the complex carbohydrates found in some East Asian diets, are found in higher proportions in the Bengali gut. This microbial signature could not have arisen by chance. It is the long-term consequence of the historical admixture between the ancestral South Asian inhabitants of the Delta and the migrating Austroasiatic and Tibeto-Burman groups. As these populations merged, they didn't just share their genes, they also shared their microbes. Through shared food, close living quarters, and the maternal transfer of microbes during birth and breastfeeding, the gut flora of the incoming and existing populations began to commingle. Over many generations, this led to the emergence of a unique, hybrid microbiome that mirrors the hybrid genetic ancestry of the Bengali people themselves. This microbial evidence provides a powerful and independent line of confirmation for the human genetic data. It shows that the integration of northeastern peoples was not a superficial event, but a deep and intimate process that reshaped the very ecosystem of the human body. The gut microbiome of a modern Bengali person is a living archaeological record, a complex community of microorganisms that tells the same story as their human DNA. A story of a people forged at the confluence of South and East Asia. In the grand tapestry of South Asian genetics, social structure has often played a powerful role in shaping the flow of genes. The caste system, with its traditional rules of endogamy, marrying within the group, has for centuries created genetic boundaries between different communities, leading to the formation of distinct, isolated gene pools even within the same village. This has resulted in many regions of the subcontinent having a highly stratified genetic landscape, where one's caste can be a strong predictor of one's specific ancestral components. However, when this same analytical lens is applied to the Bengali people, a remarkably different and much more egalitarian picture emerges. Extensive genetic studies that have sampled individuals from a wide spectrum of Bengali society, from the highest priestly Brahmin castes to the Kayasthas, a scribe caste, from landowning agricultural communities to Muslim farmers, have consistently found a striking degree of genetic uniformity. The fundamental ancestral components, particularly the characteristic blend of South Asian and East/Southeast Asian ancestry, are present in remarkably similar proportions across all these groups. A Bengali Brahmin's genome, for instance, is not significantly more North Indian or less East Asian than that of a Bengali Muslim farmer from a neighboring district. This genetic homogeneity across traditional social and religious divides is a profound statement about the social history of the region. It strongly suggests that the rigid endogamy that characterized caste society elsewhere was not the dominant force in the formation of the Bengali population. Instead, the data points to a long history of significant gene flow between different social groups. This implies a past where the boundaries between castes were more fluid and permeable, allowing for a degree of intermarriage that would have been unthinkable in more orthodox regions. This biological egalitarianism does not mean that social hierarchy was absent in Bengal, but rather that it did not translate into the creation of permanent, isolated genetic reservoirs. The shared biological heritage reinforces the idea of a cohesive and singular Bengali identity that often transcends caste and religious affiliations. The forces of cultural and linguistic unity in the region appear to have been stronger than the forces of social division, at least at the genetic level. This shared identity is not just a modern political or cultural construct. It is a biological reality rooted in a shared history of admixture and intermingling. The fact that different communities, with vastly different social statuses and histories, all draw from the same foundational gene pool is a powerful testament to the unique social dynamics of the Ganges Delta. It suggests a society where assimilation and synthesis were the prevailing currents, creating a single, relatively uniform population from a multitude of diverse ancestral streams. Before the first farmers planted rice in the fertile silt of the delta, before the first cities rose, and before the arrival of Indo-Aryan languages, the land that would become Bengal was home to a people whose legacy forms the deepest and most ancient layer of the region's genetic foundation. These were the indigenous hunter-gatherers of the Indian subcontinent, the descendants of the first wave of modern humans who migrated out of Africa and arrived in South Asia some 65,000 years ago. Geneticists refer to the ancestral component derived from this population as Ancient Ancestral South Indian, ASI, and it is a fundamental element in the DNA of virtually every person native to the subcontinent. For tens of thousands of years, these hunter-gatherer groups were the sole human inhabitants of the delta. They lived in small, mobile bands, their lives intricately woven into the rhythms of the region's unique and dynamic ecosystem. They would have possessed an intimate knowledge of the delta's flora and fauna, hunting the deer and wild boar in its marshy forests, fishing in its countless rivers and estuaries, and gathering the rich variety of wild plants, fruits, and tubers it offered. Their technology, consisting of stone tools, bone implements, and perhaps simple watercraft, was perfectly adapted to this riverine world. Genetically, these early people were distinct from the populations that would arrive later. Their DNA carries the signature of a long period of isolation and adaptation to the South Asian environment. While no pure ASI population exists today, their genetic markers are ubiquitous, forming a substantial bedrock of ancestry in all modern South Asian groups. In the Bengali people, this ASI component is a significant part of their South Asian heritage, a direct and unbroken link to the earliest inhabitants of their homeland. The arrival of later groups, first agriculturalists and then the Indo-Aryan speakers, did not lead to the complete replacement of these indigenous people. Instead, a long and complex process of interaction and admixture began. The hunter-gatherers were slowly absorbed into the emerging agricultural societies, contributing their genes, their deep knowledge of the local environment, and perhaps elements of their culture and belief systems. This ancient legacy is more than just a faint echo in the Bengali genome. It is a foundational pillar of their identity. It represents the timeless connection of the Bengali people to the soil of the delta, a heritage that predates all recorded history, all empires, and all modern social distinctions. The ASI component is the genetic proof that the story of the Bengali people begins not with the arrival of outsiders, but with the deep, ancient past of the indigenous populations who first called the Ganges Delta home. Several thousand years ago, a new and transformative chapter began in the history of the Indian subcontinent, and its effects would ripple all the way to the eastern floodplains of the Ganges Delta. This period was marked by the arrival of groups from the northwest, originating from the vast Eurasian steppe, who brought with them new technologies, a pastoralist lifestyle, and most importantly, the early forms of the Indo-Aryan languages that would come to dominate the northern half of the subcontinent. The genetic signature of these people, often referred to as Ancestral North Indian (ANI), carries links to both the ancient inhabitants of the Near East and the steppe populations of Central Asia. Their migration was not a single event but a protracted process spanning centuries, a gradual eastward and southward movement of people and culture. As these Indo-Aryan speaking groups spread across the northern plains, they encountered and mixed with the indigenous ASI populations who were already settled there. This interaction and admixture created the primary ancestral gradient that defines the genetics of South Asia today, with the proportion of ANI ancestry generally being highest in the northwest and decreasing as one moves south and east. By the time these influences reached Bengal, they had already been filtered through centuries of interaction with other groups across the Gangetic Plain. The arrival of ANI ancestry in the delta was not a cataclysmic invasion, but more likely a process of elite dominance and cultural diffusion. Small groups of Indo-Aryan speakers, perhaps as priestly and warrior clans, established political and social control, and their language and cultural practices, including elements of the Vedic religion, were gradually adopted by the local population. This cultural shift was accompanied by genetic admixture. Intermarriage, though likely structured by an emerging social hierarchy, was common enough to introduce the ANI genetic component into the local gene pool, layering it on top of the existing ASI and Austroasiatic foundations. This ANI contribution is a crucial part of the Bengali genetic makeup, typically forming a significant portion of their overall 50 to 60% South Asian ancestry. It connects Bengalis to the broad sweep of North Indian history and to the linguistic family that includes Hindi, Punjabi, and Marathi, as well as the great literary traditions of Sanskrit. The ANI imprint on the Bengali genome is a testament to the region's integration into the wider cultural and political world of ancient India. It represents the westward-facing aspect of Bengal's heritage, a legacy of a linguistic and genetic wave that fundamentally reshaped the northern subcontinent and left a permanent and significant mark on the people of the Delta. The genome of a modern Bengali person is a living historical document, a remarkable synthesis of the diverse human stories that have converged in the Ganges Delta over millennia. It is not the product of a single origin or a linear progression, but the outcome of a complex and multi-layered process of convergence, a testament to the region's role as a biological and cultural crossroads. The narrative encoded in their DNA is one of profound synthesis, where distinct ancestral streams have not merely coexisted, but have truly merged to create something new and unique. The story begins with the deep foundation of the ancient ancestral South Indians, ASI, the subcontinent's indigenous hunter-gatherers, whose legacy connects Bengalis to the first modern human inhabitants of the land. Upon this ancient bedrock was layered the genetic and linguistic influence of the ancestral North Indians, ANI, the Indo-Aryan speaking peoples who swept across the northern plains, linking Bengal to the great historical currents of the wider subcontinent. Yet, this two-part mixture, which defines much of South Asia, is only the beginning of the Bengali story. What truly sets the region apart is the immense contribution from the east. Successive waves of migration brought Austroasiatic and Tibeto-Burman peoples from the northeastern frontiers, infusing the delta with a massive dose of East and Southeast Asian ancestry. This was not a minor addition but a fundamental reshaping of the population, a demographic event that now accounts for up to a third of the typical Bengalis' genetic heritage. These grand movements of people were then overlain by the subtle but powerful forces of natural selection. In the crucible of the delta's environment, genes that offered resistance to endemic diseases like malaria and cholera were favored, altering the frequency of hemoglobin variants and blood types, further sculpting the genome in response to local pressures. The final, and perhaps most remarkable, element in this synthesis has been social. Unlike in many other regions, the diverse genetic components that flowed into Bengal were not kept isolated by rigid, long-term social barriers. A history of relative openness to intermarriage and social mobility allowed these disparate ancestral threads, ASI, ANI, Austroasiatic, and Tibeto-Burman, to be woven together into a tight, cohesive fabric. The result is the modern Bengali genome, a harmonious blend that reflects its composite origins yet displays a striking uniformity. It is a genome that is unequivocally South Asian at its core, yet carries an unmistakable and powerful echo of the East. It speaks of a history not of conquest and replacement, but of assimilation, adaptation, and integration. To be Bengali, in a genetic sense, is to be a living embodiment of this synthesis, a person whose very DNA tells a story of the meeting of peoples, the merging of worlds, and the creation of a singular, vibrant identity in the fertile soil of the Ganges Delta. In the end, the Bengali story is one of incredible synthesis. It's a tale of how diverse groups, indigenous hunters, northern migrants, and eastern tribes, came together in the fertile Ganges Delta. Over centuries, they didn't just share the land, they shared their very genes, creating a unique and unified people. Their DNA is a living testament to a history of assimilation and resilience, proving that unity can arise from incredible diversity. What are your thoughts on this genetic history? Please share any comments for improvements below.